Dear colleagues, I would like to thank the organizers for the opportunity to present this talk. My name is Arina Kichko, and I'm working at All Russia Research Institute of Agriculture Microbiology. The topic of my report is a shift in microbial community of soil in long-term burial conditions. Buried soils or paleo if the soils covered by a layer of another soil or another material in the past. Fresh organic matter is no longer delivered to the buried soil for a long time, and as well as humidity, temperature, and aeration regimes changes. Therefore, burial strongly affected the soil microbiome. It is believed that soil microbiome is partially conserved, and microbiome data allow us somehow to obtain information about environmental conditions at the time of burial. This assumption is used in archaeological investigations of burial mounds or kurgans to get some details about age and climate conditions at the time of its formation. It is known that some of morphological and chemical properties are preserved in buried soil, but on the other hand, diagenesis of soil organic matter is observed. It results in a decrease of content and changes in the structure of soil organic matter and shifts in structure of soil microbiomes. At this picture, you can see excavation of burial mound and some interesting artifacts obtained from this site. To assess changes in the prokaryotic community during burial, a comparative analysis of microbiomes of dark chestnut buried 2,500 years ago and the adjacent surface dark chestnut soil located in the same landscape was performed. To scale differences between buried and surface soil, other soil types, chernozem, sodpozol, and gray soil were taken for comparison. Quantitative PCR analysis shows a decrease in total abundance of bacteria in buried soil. The most reduction of abundance is observed in buried B horizon, and the lower reduction is observed in buried A horizon. As you can see, the most differentiation by total number of bacteria is observed between A and B horizons of buried soil. At the next step, we analyzed biodiversity. As we see in this table, both soils show the same patterns of alpha diversity coefficients distribution through profile. This factor can evidence the preservation of profile differentiation. It is interesting that B horizon of buried soil stands out of other horizons by low alpha diversity coefficients. even in comparison to the sea horizons. And buried A horizon shows higher level of phylogenetic diversity coefficient. As a plot with beta diversity, we can see that structure of soil microbiomes shifts notably. The most differences are observed in the A and B horizons of buried soil, while C horizons stay very similar. It looks like burial not affect doesn't affect the microbiomes of the deepest horizons. It is interesting that the differences in both taxonomic and abundance structure were observed. To scale different differences between buried and surface soils, we compared it with soils of contrast soil types. As you see at the beta diversity plot, differences between buried and surface soils are comparable with differences between different soil types. As box plot shows, A and B horizons of buried soil are strongly differentiated and comparable only with horizons differentiation of subpodal soil. Taxonomic analysis has revealed significant differences in microbiomes of different horizons even at the phyla level, especially for actinobacteria, protobacteria, firmicutes, taumarchiota, acidobacteria, chloroflexi, bacteroidetes, planktomycetes, and verrucomicrobia. As you can see, actinobacteria is prevalent in both soils and shows high level of diversification at a lower taxonomic level. Buried soil demonstrates different phylogenetic structure of dominance that's remarkable even at phyla level. At this picture, you can see changes in structure of dominant phylotypes in a horizon of buried and surface soils. This plot you can see abundance of each phylotype in buried and surface soil. It is clear that a small subset of phylotypes has comparable abundance in both soils. Also, as you can see, number of such phylotypes increases a bit in the sea horizons. With DSEC2 package, we have distinguished part of microbiome that significantly changes in abundance, in conditions of burial. At the basis of that subset, we construct a phylogenetic tree and color each phylotype by its dynamic trend. It is interesting that on such tree we can see that not particular phenotypes, but rather whole clades demonstrates trends to increase or decrease with burial. At this slide, you can see tree constructed for A horizons, and the next slide you can see analogous tree constructed for B horizons. As a basis of differentially represented parts of microbiome, we identify taxonomic groups which are markers of viral process. Thus, in the buried soil, a decrease in the genus Gaella, orders Rubobacterialis, Solirubobacterialis, Nitrosospiralis, Franchialis, and an increase in the acidimicrobia class Phyllofirmicutes and Chloroflexi were observed. In the upper horizons, the shares of bacteroidetes and Virocomicrobia increased. Here we show the main trend in buried soil microbiomes specificity. Cluster analysis of centroids demonstrates shifting of buried horizons microbiomes to one cluster with mineral sea horizons. It is clear sign of trend when A and B horizons microbiomes are getting closer to mineral sea horizons. These results are correlated with carbon and nitrogen content that shows decrease with burial. With the program Precast 2, we tried to reconstruct metabolic pathways from the taxonomic structure and to identify pathways with differential abundance in buried or in buried and surface soil. Results of Precast 2 and Krasko-Wallis test show increase of degradation pathways abundance in buried soil. At this heat map, you can see pathways with significant shifts in A horizons, and all degradation processes are marked in blue circles. Cluster analysis of microbiomes together with carbon and nitrogen content data and results of Precast 2 are clearly showing a trend to overall mineralization of buried profile with its gradual shift to a state of mineral sea horizons. And our conclusions. Buried profile keeps stratification by morphological properties, number of bacteria, and structure of microbiome. Burial significantly change soil microbiome, and the scale of these changes is comparable with the differences between soils of different types, climate, and vegetation. In the buried soil, a decrease in the genus Gaella, orders Rubobacterialis, Solirubobacterialis, Nitrosospiralis, Franchialis, and an increase in the acidimicrobia class, Phyllofirmicutes, and Chloroflexi were observed. In the upper horizons, the shares of bacteroidetes and Virocomicrobia increased. Horizons of buried soil demonstrate some trend to overall mineralization, as evidenced by increase in part of degradation pathways negative dynamics of carbon and nitrogen currents, and clustering microbiomes of buried horizons to the same group with ones of mineral-sea horizons. However, microorganisms involved in the cycles of biogenic elements still exist in buried horizons. Among them, Nitrolanthia, Candidatus alizosphaeria, Bacillales, and Candidatus nitrososphaeria for nitrogen cycle. Thank you for your attention.